Next, we want to talk just briefly about red blood cells and how they transport oxygen. So our red blood cells, they're also known as erythrocytes. These have a very interesting shape. They have sort of a biconcave shape. They kind of look like a donut without the hole actually punched out, but just caved in. But guess what? Guess what happens when you do that? You are massively increasing your surface area to volume ratio. A biconcave disc has significantly more surface area than a perfectly round sphere. Has a significantly greater surface area to volume ratio than a sphere does. And so this is really, really important, especially when you consider red blood cells' primary goal, which is to transport oxygen. So it does this by the protein that it contains, which is hemoglobin. Hemoglobin is a pigment that binds to oxygen, and it's the reason that red blood cells are, and thus blood are actually red. The heme portion of hemoglobin binds up to 4 oxygens. It can also bind carbon monoxide, and this is the dangerous part. Hemoglobin actually has a preference for carbon monoxide as opposed to oxygen. This is why carbon monoxide is so dangerous, because your blood cells will preferentially bind to carbon monoxide, which it cannot use for the cellular respiration. It desperately needs oxygen. And so that's the danger of carbon monoxide, is that your blood cells can bind to carbon monoxide instead of oxygen, thus slowly starving and depriving your body of the oxygen it so desperately needs. When bound to oxygen, hemoglobin is called oxyhemoglobin. When oxygen leaves hemoglobin in the tissues, it's called deoxyhemoglobin. So hemoglobin comes in 2 forms, that bound to oxygen and that unbound to oxygen. The production of red blood cells. Now, this one's really interesting because red blood cells are going to be produced in your bone marrow. The red bone marrow specifically. We'll talk about yellow bone marrow a little bit later on when we talk about the bone structure and function. But as red blood cells are produced, they are ultimately going to lose their nucleus and most of their organelles. They start out looking a lot like most of our other animal cells, most of our other eukaryotic animal cells, the nucleus and mitochondria and the Golgi apparatus and endoplasmic reticulum. But over time, they're going to lose their nucleus and those organelles. Without a nucleus, the cell cannot make proteins for cell repair. We desperately need our nucleus. Our nucleus contains all of our genetic information for making more cells, making the proteins that cell needs to function. Because these red blood cells do not have their own nucleus, they're going to have to be removed from the system. So red blood cells only live about 120 days. After that, the old worn out cells are going to be removed from circulation by macrophages in the liver and spleen. The disc shape allows them to squeeze through the very small capillaries, and it also allows for maximum surface area, like we talked about in the previous slide. The reason we care about that maximum surface area is because it carries our oxygen and our carbon dioxide. The more surface area we have, the more efficient those cells will be for the diffusion of our gases. So erythropoietin, EPO, it's a hormone that's produced by the kidneys when oxygen levels of the blood are low. This is going to stimulate the bone marrow to produce more red blood cells. Blood doping, you may have heard, happens when you increase the number of your red blood cells, sometimes even by injecting EPO. So erythropoietin, that hormone, can be injected into the bloodstream to stimulate the production of more red blood cells. This has been done to help increase stamina and athletic performance. With more red blood cells in the body or in the bloodstream, the more potential there is for carrying oxygen. Oxygen is what our cells need for cellular respiration to produce that chemical energy ATP. The more red blood cells we have, the more oxygen we have, the more our mitochondria are able to produce ATP. This is incredibly dangerous. I am not recommending this. Blood doping is incredibly dangerous. A lot of times what happens when you have too many when you have too many red blood cells in the bloodstream, it can actually cause your blood to thicken And when it becomes too thick, it actually won't flow. If it's not flowing correctly, then your body is not getting the oxygen that it needs, thereby defeating its purpose. And this can cause heart failure in the long run. So it's very dangerous. Your body produces its own erythropoietin when needed, as needed. There's no need to blood dope by adding more erythropoietin to the bloodstream. There are several disorders that involve red blood cells. Jaundice is an accumulation of heme in the blood if the liver can't excrete it. What happens is that the skin and the whites of the eyes can start to turn yellow. Anemia happens when we have too few red blood cells or too little hemoglobin. So ultimately, anemia is caused by a lack of oxygen, and again, due to too few red blood cells or not enough hemoglobin in those red blood cells. There are a couple of different types of anemia. Iron deficiency anemia, this is the most common form. This is caused by an inadequate intake of dietary iron. So you can't make the hemoglobin without the iron. You can't make the hemoglobin without the hemoglobin. You can't transport as much oxygen as is needed. There's also pernicious anemia. This is where you have a lack of vitamin B12, which is needed to make the red blood cells. So in this case, there are too few red blood cells. The hemoglobin levels are fine, but there's just not enough red blood cells to carry the oxygen. There's also folic acid deficiency anemia. You also need folic acid to produce red blood cells. So without it, there's too few red blood cells, not enough oxygen. Hemolytic anemia, this is caused by too much hemolysis. So the red blood cells themselves, there may be enough red blood cells, but they keep rupturing. They're very weak. They have weak cell walls. They keep being damaged and rupturing. If they do not have their correct shape, that biconcave disc with that plasma membrane on the outside, they're not able to transport oxygen. And so we get hemolytic anemia. Sickle cell disease is a genetic disease that causes red blood cells to become sickle-shaped and prone to rupture. So here is that sickle cell-shaped red blood cell. Healthy red blood cell has this sort of round biconcave disc. A sickle shape has, of course, a sickle shape. The danger to this is that they become very fragile, very brittle. They're prone to rupture. This is caused by a defective hemoglobin structure. Hemoglobin, like we said, is a protein. Protein structure and function is determined by our genetics, determined by our DNA. And so this is how that genetic disorder can cause that sickle cell shape. We'll talk about this quite a bit more when we talk about our translation from DNA into proteins and when we get into our genetics a little bit later on in the semester. All right, so just to review this section, you should be able to explain how red blood cells transport oxygen and carbon dioxide by outlining the chemical processes involved. Really, I just want you to focus on the structure of a red blood cell, how that makes it well suited for gas transport. We did not spend a whole lot of time on hemoglobin structure, and I'm not going to ask you for that information. I just want you to focus on that biconcave disc, focus on the fact that red blood cells do contain hemoglobin, which is going to be our oxygen carrier. And then also you should be able to describe the role of EPO in red blood cell production. We are going to talk about hormones a little bit later in the semester, so this will come up again, which is why I'm encouraging you to review it and go over it now.